From shallow water reefs to the mysterious deep, OceanX is on a mission to explore the ocean and bring it back to the world. Here's a look at the groundbreaking research conducted on OceanXplorer in collaboration with researchers from the University of Bergen.
Jellyfish are often ignored. They are one of the sort of original predators. Can we film it? Yeah, we want to film that. So it's very exciting to see what we find. We have some good guesses, but whether we will see those species, we will see. Anyone who has ever seen pictures or videos of jellies knows that they are super beautiful. They are extremely photogenic, and this is not what we normally get in the nets. So it's wonderful to be able to film them in situ using ROVs or submersibles, and to see them in situ, and also to collect specimens in good condition. It's a unique opportunity to see deep water jellies as they're supposed to be seen. My name is Aino Hosia, and I studied jellyfish diversity and ecology. I moved to Bergen in Norway to do my PhD in 2003 and have been working with jellies ever since. There are two things that are challenging about studying jellies. One of them is sort of convincing people to actually also study the jellies, because people always just want to study the crustaceans and the fish, and then jellies are sort of forgotten. The other is they're very fragile. So using traditional nets is not the best method to study them, because they will get squished in the nets and they will be broken to bits and pieces, and you won't get a good idea of the diversity. Understanding biodiversity is key to protecting ecosystems, because in the ecosystem everything is connected to everything. Without understanding what species there are, what roles they play, then you cannot really understand the entire ecosystem. And jellyfish are a component that are often ignored, even though they are very important for the system as major predators. They are one of the sort of original predators, so they're an extremely successful group that has been around since prehistoric times. I mean, much longer than dinosaurs or sharks or other prehistoric animals that you would imagine. And they have been pretty much unchanged since then. They are very successful in what they do. The gelatinous fauna of the Norwegian Sea is actually not that well known. So it's very exciting to see what we find. We really don't know. We have some good guesses, but whether we will see those species, we will see. It's an aglantha. It's a little Trachymedusa, a hydrozoan jellyfish. So while we're here in OceanX, we plan to do several different things. First of all, we're using the filming opportunities and the collection opportunities that we have with the ROV and the submersibles to collect these very fragile animals in good condition. Oh, that's nice. Oi. Because we've brought with us our net, so we can get net samples from the whole water column and see what's there. We can go through it physically and identify things morphologically and count them and see how many of each species there are. Then we are doing the vertical transects with the ROV, where we can also identify the species visually and see where in the water column they are. And in addition, we're taking water samples with the CTD. Gelatinous zooplankton, it has a lot of different definitions. What we work with is gelatinous predators. So it's gelatinous animals that feed on other zooplankton mostly. And that includes different kinds of jellyfish, both the large scyphozoan jellyfish like the moon jelly and the lion's mane jelly. But most of the diversity is actually in a group called hydrozoans, which are very small jellyfish. In Norway we have more than 100 species. It includes tiny jellyfish, and it also includes this very cool colonial jellyfish called siphonophores. And most people may not be familiar with siphonophores, but most people know at least one siphonophore, even if they don't know that's what it is, and that's the Portuguese man o' war. And then it includes ctenophores, or common jellies. There might be some cool species because we have both the Arctic water and the Atlantic, so you can have all the krill you want. We have been working on DNA barcoding the Norwegian fauna of jellies. You use a short sequence of a specific gene, and this is a gene that is very good for separating different species. So if you sequence it and you look at the sequence and you group these, then you will be able to separate the species based on this gene. And we're building a reference library, sequence library, which allows identifying the animals just based on their genetics. Ooh, that's nice. I think we have most of the commonly occurring species of gelatinous zooplankton in Norwegian waters in our database. So we are lacking few of the rare ones, which we hope that maybe we'll encounter here. The thing we're testing is just called eDNA, or environmental DNA. When animals are swimming around in water, they are all the time losing cells. Lots of jellies, we'll see. Thank you. And then this DNA is in the water column. Just by taking a water sample and filtering all the DNA out of this and then sequencing the DNA that we filtered out, you can get a sort of a list of species that are occurring in the area, theoretically. Yes. And there are some mysids like here floating, I think it's dead though. Yes. And some super large copepods. Jellies, because they're so fragile, they're very often ignored in monitoring. That's why we would also like to have alternative methods for monitoring the diversity of gelatinous zooplankton, because it is still a considerable portion of the total diversity of plankton. And of course they're predators, so they're ecologically very important.
I think you can try with both?
I think it's in now, yeah.
Wow, that's a big colony.
I think we need some more water there.
Yeah, okay.
During the past 20 years, it has been more and more accepted that jellies are a very important component of the marine ecosystems, both as predators and as prey. The previous view was that jellyfish are dead end, they're not energetically good to eat because they are 99% water. But then again, jellies can be very large, and that means that they can also be easy to find. So still, if you find a single jelly, that can be a lot of food compared to a tiny copepod. Also, jellyfish contain lots of different parasites. So it's actually like a concentrated buffet. It's not just a jellyfish, but it's everything else that lives within this jellyfish. So it might actually be very good food as well. And that's what we're finding out, that jellyfish do form an important part of the diet of many animals. Different species of fish, several seabirds, sea turtles. Nice. Oh yeah. That was actually, I think it was probably Albatictina. That's not something that we often see. We're becoming more and more aware that we're destroying a lot of biodiversity, both terrestrial diversity and marine diversity. Jellies, they have been often ignored in monitoring, so that's a large part of the diversity that we know very little about. And if we know very little about, then we also can't see the changes in diversity. We don't know if we lose species or if the species composition changes, because we don't sort of have a starting point to compare to.
It's a...
Oh, it's a...
Can you say this and stop?
Can we film it? Yeah, we want to film that. Oh, that is so cool. The oceans are by far the largest habitat on Earth, and we still know extremely little about them. Which is why the kind of ocean exploration that OceanX provides opportunities for is extremely important.
Yeah, Roger, Chris. I am on bottom, depth 395 meters.
What is that small one right there?
What's that?
The small one right there?
That is a lantern shark. That is one. Oh my god. It's coming. No, stay, stay. My name is Nicholas Straube, and I'm an evolutionary biologist studying deep-sea sharks. I'm joining the OceanX expedition for trying to film the bioluminescence of the velvet belly lantern shark in the fjords of Norway. Shark bioluminescence has never been observed in the wild, and this is something we intend to do during this expedition. Since I'm a child, I'm interested in sharks, especially deep-sea sharks, critters with huge eyes and special features. The velvet belly lantern shark is very abundant in Norwegian fjords, and it also occurs here, in contrast to the open Atlantic Ocean, in rather shallow depths. So I take the fish book. Good idea. Given those two things, high abundance plus easy accessibility, this species became a model species for the bioluminescence research.
Yeah, Roger, we're happy for divers in the water. I like this right here. Then as we go down, we can turn into the sun. Perfect. I'm ready to bet.
A general definition of bioluminescence would be the capability of organisms to emit light. Bioluminescence is very common in the deep sea, and given we have more than 500 species of sharks on Earth, about 11% are actually bioluminescent. Confirmed in terms of seeing them glow, it's only about 5 or 6 species. I don't think too many people are aware of that.
So this is Neptune venting now.
Roger, venting now.
The bioluminescence system in sharks is still very poorly understood. They have these photophores. A photophore is basically the organ which emits the light. In lantern sharks, this is a combination of different cells embedded in their skin, and that is known for quite a while now, since specimens were collected for scientific fish research since hundreds of years. If you analyze the skin of those specimens, you will see the photophores. 15 years ago, research started on analyzing this bioluminescent system. And experiments showed with live animals that they actually glow.
Control, this is...
The bioluminescence serves likely different functions, but we never observed that in the wild. One function which is kind of very likely to be fulfilled is counter shading, which is a type of camouflage. Imagine that you're a predatory fish who is living in the deep sea and is looking up to the surface. And if a fish would pass by above you, you would see a shadow. This is actually a lantern shark with lights off. So you would clearly see there is potential prey. But as soon as the shark switches on its light, it disappears. And that cloaking of its silhouette is called counter shading. A lot of different deep-sea taxa do that. It seems to be very important for survival. Okay, so this is actually a lantern shark specimen from the fish collection from the Bergen Museum. They have these lateral markings. We call them flank markings. So what we can see here is the flank marking. Those markings are believed to be useful for schooling, so that they can stay together in groups. And we can also see one of the dorsal fin spines, which is right here. Those spines are illuminated as well. This could be used to just tell a potential predator, don't bite me or you will be pierced by these spines. Their back also carries photophores, but it does not light up as bright as the belly. It's just certain patterns, stripe-like patterns, which may be used to warn off predators. It's just 40, 50 centimeters. So if you're that size, there's numerous fish which could eat you.
And visibility lights on, 10 meters, maybe 15, over.
We will use the gear OceanX provides, submersibles and ROVs, with the sufficient camera equipment to try and film the bioluminescence of the lantern shark in the wild. A lot more going on compared to last time. Ideally, we would be able to support or falsify the hypothesis assumed for their different functions.
This is it. This is lantern shark territory, right? That's all this time.
I'm certainly nervous that we don't run into enough sharks to film the bioluminescence properly. We really have to meet them by chance.
What is that small one right there?
What's that?
The small one right there?
That is a lantern shark. That is one. Oh my god. It's coming. No, stay. Fantastic.
Okay, great. Well, we got some.
But he was biting the boat. You see that? Yes,
Oh, here
he was biting the
I think
boat.
he likes our bait.
Okay, I think we should turn lights off. Yes, please.
So we went diving in Fjord, trying to film the lantern shark bioluminescence, and we were seeing several, and we do have a little footage, at least in bright light, of a lantern shark, which I think will turn out very nice. And yeah, and we have to review the footage. I'm really excited. That was the first time actually I saw one alive. And especially since I normally work with museum specimens, seeing them live is fantastic. They are very cute and very beautiful animals to me. That's a very nice footage, yes. You can see the black belly. Wonderful. All the very dark areas, for example, the belly, are covered in high density with phosphorus. Oh yeah, here it comes. Very slowly swimming in mid-water, right above us, but you cannot see any luminescence here. I wonder if the remaining light from the other side may be too bright, actually, because the light is probably very dim, but I can see a clear shadow. Huh. I would expect that it would light up. Okay. No luminescence here, unfortunately. Well, next time.
So you're not discouraged?
No, never. The opportunities to film them in their natural habitat alone is fantastic. We saw it's possible to meet them down there, and we know now how they behave and how to film them. I would see those results as part of understanding the ecological role of lantern sharks, which is also still poorly known and understood. And the more we know about that, the better we are able to manage and conserve species like that. The subdive was fantastic, I have to say. That was a dream come true.
Coming back to that. He's coming right to you. Oh boy.
Oh man,
Oh boy.
that is fantastic.
Over the last 50 years, kelp forests around the globe have been degrading. And that is in part due to a lack of protection and in part due to environmental changes. There's a variety of different threats. One of them is increasing temperatures. I think if we were able to show the natural beauty of kelp forests to people, then that would help a lot for us to actually protect them. On this cruise, I am studying kelp and rockweeds. My research here will be part of ongoing efforts around the globe to better understand the role of kelp in carbon sequestration and climate change mitigation. What we would like to do here on board the OceanXplorer is to look at the different kelp species that we find along the stations, along the coast, and take samples of them and analyze them for their carbon content.
For each species that we find in this location, I would like three from three different spots.
Yes.
And then, a few pieces, basically.
Technically, kelp, they don't belong to the plants. They are macroalgae, so they are more primitive in a sense. It's a multicellular organism that you find in nearshore areas, and they essentially grow through photosynthesis. You find them around about 25% of coastal areas around the globe. Kelp can grow very big, and when you dive through them, you get the experience of being in an underwater forest. And what makes them special is that besides the sheer size of them, they provide a habitat for a lot of different species. So they are very important for fish nurseries, and for biodiversity. They provide a lot of food for a wide variety of different species, and they provide protection against erosion. They serve a lot of different ecosystem functions. And because they can take up so much carbon, they actually play an important role in the global carbon cycle. 40 to 60% of carbon taken up by plants around the globe is being taken up by the ocean. If the carbon that they take up is being stored for a long time in the ocean, it is considered to be removed from the carbon cycle of our system, and thereby actually helps to mitigate climate change. But in order for that to actually work, kelp needs to stay in the ocean. And if it gets buried in the deep sediments, that's when we have a fairly good idea that it will stay there for a significant amount of time, for hundreds of years. The problem is that we don't really know how much is being essentially transported to deeper areas. That's in fact the reason why we're here. Besides taking the samples of the actual plants, we also use drone footage in order to understand better the extent of the kelp forest that we are looking at. That will help us, of course, to get a better idea of how much kelp there actually is. And then we do take sediment cores off the stations where we look for kelp and look for kelp-specific markers in the DNA that we find in the sediment. So that allows us to not only see if there was a specific kelp species, but also to some degree quantify the amount of different kelp species in the sediment. We do need to study kelp, because only then will we be able to implement policies and regulations that allow us to protect kelp. It is vital that we get a better understanding of how the ocean works in all its different shades and colors. It is by far the largest ecosystem on our planet, and it is just so crucially important for the health and for our bare existence on this planet.
Oh, that was a flash. A white flash. Der var noter.
Yeah.
But that was definitely a flash.
It is amazing how little we know about the deep sea. It's not very well investigated because of the challenges by going into the deep. But we're getting closer now, especially with the ROVs and the fantastic new submarines.
My name is Henrik Lenner.
My name is Anders Garm.
On this mission, we are going to work on bioluminescence.
The understanding of the deep sea is extremely important because it's a huge environment. The deep sea ocean floor covers maybe half the Earth's surface. If we really want to understand the diversity of biology, the diversity of life, our own evolution for that matter, we definitely also need very much to consider what happens in the deep sea.
There's a lot of stuff that is completely unknown. One of them, of course, is why are most of these animals we find down there, why are they bioluminescent?
Bioluminescence is light produced by living organisms. Classic example is the fireflies. It's actually one of the most common traits of animals in the sea. 3/4 of all animals from the surface to the deep sea are bioluminescent.
And when you have a thing that is so common and has evolved several times, then it must be super important. And that's why it's important also for us to find out why.
In 99.999% of all the cases, we don't really know why they do it.
We are going to investigate a certain organism.
They're called brachyngid starfish.
It is a starfish that is relatively large.
The biggest one goes up to 80 centimeters in diameter, so almost a meter. The common starfish, will typically have 5 arms. The ones we'll be looking at will have between 10 and 12 arms.
On certain areas of Sognefjorden, they are one of the dominant invertebrate species on the seafloor. It's a fantastic situation where you have this relatively rare organisms in enormous amounts.
We know very, very little about them because they're almost only found in the deep sea. But all the 5 species of bracyngids we have tested, they are all bioluminescent.
We don't know why they are bioluminescent. We just know that they're able to send out this light.
Just like a lot of other, especially deep sea animals, we have only just a faint understanding on how these animals function. Ja, det var dejligt. Starfish are actually pretty complex animals, even though they do not seem to be. We discovered a few years back that some of them have really good eyes. Now, eyes in starfish is somewhat of a surprise for many people, but they have eyes pretty similar to the eyes from insects. Our idea is that they use these eyes for communication. So down there on the seafloor, they might be flashing at each other and then seeing it so they can communicate things like, I'm a male, I want a mate, are you a female? So they can sort of find each other in that way. So that's the hypothesis, one of them we're going to test on this cruise. We're testing 2 other. One is as a prey lure.
They are extremely slow animals. They are not able to move very fast after prey. The way you see them in the sea is that they lift their arms above the seafloor.
They sit like a basket, so the arms sort of fold up, and they feed as sit-and-wait predators.
Then they are sending out this light.
A lot of animals living in the dark are attracted to light. So you know it from at night if you have your light on your porch, and you will have all kinds of insects coming in, swarming around your light. A lot of animals in the sea does the same. So another hypothesis is that when they do these small flashes, copepods will come in and be curious and attracted to this.
And then they are trapped by the arms.
The last hypothesis we're also testing is what we call the burglar alarm, and it's sort of going by the theory that the enemy of your enemy is your friend.
When a fish is coming, eating, or trying to bite something of the organisms, they are sending out strong light. And this strong light is causing other larger predators to come, and the larger predators are eating the smaller predator.
And thereby sort of rescue the starfish.
So that might actually be what is happening with our starfish.
Oh, it's a sea star.
Yeah, it is.
Yes, let's pick it up.
Look how beautiful it is. I mean, it's wow. What we want to do is to look at the behavior down there. It has never been recorded in the natural environment.
We'll be able to send down really sensitive cameras and film the bioluminescence on the bottom at the depth of 1,200 meters. We'll collect the animals and then we'll do further experiments in the tank in the lab. was a flash, a white flash. Wow, der var noter.
Yeah.
But that was definitely a flash.
It's a fantastic opportunity to simply use a robot arm to collect them. They are fragile, so we have to be very careful.
Bioluminescence, it's a strange phenomenon that sort of is not part of our own daily lives. But what is a part of our daily lives is light. We're always really fascinated by light. We know that from art, we know that from culture, we know that from just being human beings. What you have to sort of realize when you're talking about bioluminescence, it's the light as such is of course interesting, but what you do when you produce light is that you want some eye to see it. It's geared for an eye. So the way eye works today has been formed by the kind of bioluminescence that has been around in the sea for a large part. And eyes might have started there for many animals to detect the bioluminescence.
So we are interconnected and we have to understand the connection. Yeah, so what actually you see here in mind is it is actually feeding. It's trying to feed.
Yeah, that could definitely be happening. Oh, there was another flash there, so that's going to be in this region here. Every single flash is going to be analyzed in detail. Intensity and color and duration.
Biodiversity is important. We know that simply the number of habitats and the organisms that have found these habitats stabilize the ecosystem. If we take away some of the species and even some of the ones that does not look like very important, a stable system can suddenly collapse.
By understanding the biodiversity, we will understand how evolution has created all the different kinds of life we know, including ourselves.
Life is coming from the sea. That's where it all originated. I think it's super important to find out for us understanding life on Earth, what is going on in this sea that we are all coming from.
This group on this vessel, we are the first people to see this specific place. It's a little bit like being one of those early explorers, when you had a white map of the world and you could go there and you could sort of say, well, this is something that nobody have ever seen before. My name is Anne-Helene Tandberg and I am a crustacean taxonomist. Aboard the OceanXplorer, I am going to study some places from an expedition 150 years ago. The historic expedition that I want to sort of follow in the footsteps of, or ship steps of maybe, was called the Norwegian North Sea expedition. In Norwegian, that would be Den norske Nordhavsekspedisjonen. The Norwegian North Sea expedition was the first scientific exploration of what we today call the Norwegian Sea. This expedition started in the south, so right outside of Bergen, and it ended north of Svalbard. They spent 3 summers in 1876, 1877 and 1878. They studied how deep the ocean was at different places, how cold or warm the water was, and how fast the water currents moved. And they studied the zoology of the area. So it's really much the beginning of science at what now is the University of Bergen. At the time when this expedition started, Most people didn't know anything about the deep sea, and a very few researchers knew that there would be life in the deep sea, and this was a thing that was not really agreed upon just 30 or 40 years before this expedition. At that time, people thought that everything below 500 fathoms was a dead area. Nothing could live there. Then the next 20-30 years, more researchers started to investigate the deep sea. So the methods that these people used 150 years ago were very different from what we can do today, especially how we do it on this expedition. This vessel has some really cool ways of looking down into the ocean, which obviously people couldn't do before. They used dredges and trawls, nets that you sort of hang out behind the boat with a long rope so that it reaches down to the seafloor. Just a couple of hours before they would deploy their nets, they would find out how deep it was for the first time. And then they would just plop down the net, pull for a couple of hours and see what was there. So in a way it was sampling very blind. Now, using this vessel, we can send down an ROV, or we can send down a sub where we can sit inside and look. So we're not blind anymore. I'm really amazed with how many things that they found using such simple equipment. 150 years ago, what they did was making beautiful drawings. Now we can have film of all we did when we sampled, and we can take photographs of the animals when they come up and they are still alive. In addition to looking at what the animal looks like, we can also look at the genetics, the DNA. These sequencing of specimens from museum collections, together with sequencing things that are brought into museum collections now, are making all that material that is there even more available to modern science. Specimens from the old expedition are still used quite a lot. I looked at specimens from this expedition when I did my PhD. I study tiny crustaceans that live on the seafloor. The group that I love the most is called amphipods. Many of the species that I'm working with now, nobody knew that they existed before this expedition. And some of them nobody have seen since the expedition. Right now, we are at a station that was visited in 1876 on the 30th of June. After that, I don't think anybody has been here. And now we see some of the species that they saw. Amphipods! These are scavenging animals, and I would say they're almost like the renovation workers of the ocean. They eat whatever is dead until there isn't much left. There was a lot more life than it seems, right? So when you zoom in a little bit on that sediment, it's all of a sudden teeming with life, and it looks very barren, and there are some black sticks sticking out of it, and then when you look more, there's all these different things living on it, and I think that's just amazing. The station that we're visiting today is a place where a lot of species were described for the first time. They had what we call type locality. Type locality is when a species is described scientifically, you need to tell where you first found it. And that needs to be written down in the publication that describes that species. One of the species that had the type locality from the station we visited is a species that I have been specifically working with the last year. The type specimens, the first specimen that were used for the description, have been lost. We need to find something back from the exact same spot where they first found it. And I did find one specimen. So this is a specimen. It is Haliragus quadricuspis, and that is an amphipod. Since we collected it with the very nice ROV, it still has all its long legs and antennae, and that never happens if you take it up with a trawl. These sites that we are visiting right now have never been visited with this kind of equipment before. So nobody have been down with either human eyes in a sub or eyes from an ROV to see what it looks like down here. And I think also seeing things in general gives you a better appreciation for the entire system. And I would wish that more people could be able to see, because I think the deep sea is so beautiful. I want to share it. One of the reasons why we should visit the sites from the Norwegian North Sea expedition right now is that the world is changing very rapidly, and we will not be able to say anything about nature today unless we understand how nature has changed. Learning about history can also learn to predict a little bit what will happen with different scenarios in the future. So it's not just understanding what has been, but preparing for what might come. I am hoping that the work that I am doing here now this week will remind people about the Norwegian North Sea expedition. I think if anybody look back at my work in 150 years time from now, I hope they will say, ooh, cool animals, I also want to look at amphipods. And it would also be very, very nice to be part of a group of people who could inspire new generations to continue with learning about the biodiversity of the sea.
The mesopelagic zone, also called the twilight zone, goes from between 200 and 1,000 meters depth. We have not that much of species diversity, but we get tons of mesopelagic fish. To understand how I want to study it, you need to understand how these fish move.
My name is Anne Gro Vea Salvanes, and I am a professor at the University of Bergen, teaching marine ecological field methods. When I came to Bergen, it was a period with too little field work for the students, so then I happened to say that when they are graduating in marine biology, they can't do that only on paperwork. And then the head of the department said, okay, make a course then. 25 years I've been teaching. One of my PhD students is on board. She is very enthusiastic and motivated.
Masfjorden is a fjord that has been extensively studied by the University of Bergen, and especially Anne Gro. She's been going there every year with a student cruise for at least 10 years.
I did my PhD in Masfjorden, and so I knew the fjord, and I knew that, okay, it's 6 hours from Bergen, flat sea, we don't get seasick. If we want to study dynamics and development and progression in the ecosystem, then we have to go to the same place where we meet the same populations.
Once we close the hatch, we're taking our 1 atmosphere with us, so you won't notice the pressure change going in the water.
We've been there consistently since 2011, and we have gotten a lot of mesopelagic fish since the beginning.
The mesopelagic zone, also called the twilight zone, goes from between 200 and 1,000 meters depth.
On my field trips, we use acoustics to spot the densities of the mesopelagic fishes.
This is like probably the coolest thing ever. We send sound, and then anything that has a different density than water reflects that sound back. Basically, when we send sound and we get the reflection, we get a picture of sound.
I'm trying to put biology into her work.
I'm interested in how the mesopelagic community interacts with benthic organisms. Benthic communities are anything that are on the seafloor. So I'm planning to study, is there energy moving from the mesopelagics to benthic organisms? More specifically, big predators such as benthic fish. To understand how I want to study it, you need to understand how these mesopelagic fish move. Mesopelagic organisms actually conduct diel-vertical migration, which is the biggest migration on the planet, and it happens daily. What they do is they feed in the upper layers at night, and when it's dark, and then when it gets daylight, they migrate down and escape from visual predators. Since they eat at the top and then they migrate down, they move energy from the top of the water column to deeper depths. Does the energy then further travel by direct contact to benthic predators? The idea is that if they meet a bottom that is in their way, for example, the side of the fjord, they don't move away, but they just get trapped there. And the question is, how much energy flows that way? The more we look at it, the more we know about it. That's why I'm really happy to be on board this ship, because there's 2 submersibles and there is 1 ROV. And the camera footage will give me a good idea if they're actually there or not. And if I'm lucky, I will also see the interaction maybe on the footage. We will be running a transect, so a line of data collection with cameras, and then also making a sound picture.
This is really very high-tech compared to what I'm used to.
I always like to think about it like this. When you walk through the forest, you have like an idea of the 3-dimensional space you're in, and you can see far away, you can see to the next mountain. But if you would put your head underwater, there is not that far that you can see. So we rely on equipment like cameras or ROVs to get an understanding of what is there.
Fieldwork is essential for being a good scientist in marine science. It's my opinion that you have to go to sea to learn.
I think I grow so much when I do field work. Like, you just have to be more flexible in your thinking, and I love challenges.
The more I learn, the less I think I know. That motivates my work. That's why I love to be teacher and doing research.